Is it Tuesday?
I don't remember.
Yeah. Are all your finals on Monday and Tuesday?
Yeah.
Cool.
I think that. Well, I thought one was on Wednesday.
Do you really? It happens. My senior year as an undergraduate, my last semester, I had 3 in a row, and I just said, screw it, I'm doing them all, I want out. I survived. It wasn't fun. What? That again, we'll go through a few more, and then I want to talk some more about sex determination. Okay, so here's another human pedigree. Notice there's several examples of consanguineous relationships in there, matings between genetic relatives. Those are typically indicated by a double line. So there's a consanguineous relationship here between, looks like first cousins. And then here's a consanguineous relationship. Not sure what they'd be with relation to each other. This is a niece of this individual, it looks like, or a great niece or something. It's something. It's consanguinity. Yeah, yikes. Okay, so the first thing that you want to ask yourself is this consistent with a dominant trait? Right? You look at the rules and you say, okay, dominant traits ought to appear every generation. And this one doesn't seem to appear every generation, although you got to be a little bit careful there, because this relationship, you could sort of take these individuals and just stuff them up back in that last generation because of the difference between these two. This one's in the previous generation. This one's in generation 4. Okay, so it's sort of consistent with a dominant trait. It's obviously not recessive, right? I mean, that should be pretty clear. Recessive traits don't usually appear every generation. Okay, so is it X-linked or is it autosomal? It's autosomal, probably. Yeah. The X-linked thing, the thing you want to look for with X-linked dominance especially is the offspring of affected males. This affected male ought to transmit the trait to every single one of his daughters because they have to inherit his X. Okay, and since he doesn't, since there are some daughters there that are unaffected, then you know that this is not likely to be an X-linked trait. The other thing that tells you that is that with X-linked traits, females are twice as likely to be affected than males. So in a pedigree with a reasonable number of individuals like this, you ought to see twice as many females affected, but you don't. You've got 4 females affected and 3 males. Okay? So again, those 2 pieces of information indicate this is not likely to be an X-linked trait. The real telling one is the offspring of this male. Okay? He should produce all affected daughters and no affected sons because they inherit his Y, all right? And yet he's got affected sons. So this must be autosomal. Okay, let's look at this one right here. Okay, dominant or recessive? Dominant. Yeah, I heard a few people say recessive. The reason it's not recessive is because it appears every generation. Recessive traits don't usually do that. Okay, what happens with recessive traits, if they're not X-linked, is you tend to see that they don't appear for several generations, and then suddenly there they are, often because of a consanguineous relationship, reproductive event between genetic relatives. All right, so this is dominant. So if it's dominant, then the next thing you should do is you should be looking at the offspring of affected males and asking, is it consistent with X-linked? Offspring of affected males. If it's X-linked, this individual should produce all affected daughters and no affected sons. Same thing with this individual. Okay, you've got two independent lines of evidence there that are consistent with that. Right? All the daughters of these two affected males are affected themselves. None of the sons are. Okay? So that's a simple way to determine, once you've determined dominant versus recessive, that's a simple way to determine X-linked versus autosomal. Look at the progeny of affected males. So this one is consistent with an X-linked dominant trait. Okay, how about this one? Okay, so the first thing you do is determine dominant versus recessive. It's clearly dominant, yes. Then you look at the offspring of affected males. Okay, if it's X-linked, affected males should produce all affected daughters and no affected sons. Therefore, because of this, right here, this cannot be an X-linked trait. Cannot be. Right? This is an autosomal dominant trait. This is a little unusual, having all of these offspring being affected. But that can happen. I mean, the probabilities for each of these individuals is independent. Just because your first child was affected doesn't mean you suddenly have a reduced probability of having another affected child. It's the same as before. So here they just beat the odds. Okay, what about this one? Okay, this one's pretty classic. Dominant or recessive? Recessive, yes, good. Okay. It's pretty obvious at that point. Okay. With X-linked recessives, males are far more likely to be affected than females. And that's exactly what we see in this pedigree. Also, we see that we have a sort of a carrier situation. This individual up here in the second generation is affected. Okay? Therefore, his mother must be a carrier. Her daughter is a carrier here. This daughter is a carrier. This daughter is a carrier. And bingo, they have two affected sons. Same thing here. This individual is a carrier, and she produces approximately half of her sons being affected. Classic X-linked recessive. Okay, so that one's pretty straightforward. Most of the time when you actually look in the literature, in clinical genetics journals, for example, you don't see pedigrees that are this clear-cut. Okay, how about this one? All right, this one's pretty classic too. Dominant or recessive? Recessive. Yeah. Probably autosomal. Okay. You have no sign of the trait until you get this consanguineous relationship, these genetic relatives, first cousins that reproduce. They produce a whole bunch of offspring, and 3 of them show the trait, but those 3 are identical triplets. See that line that comes down and then it splits? That indicates one reproductive event, one zygote that's split to form 3, and so these are identical triplets. No wonder they all have the trait. Okay. The lines that are through here, these, if you see a line, a diagonal line through an individual, that indicates that the individual is deceased. Okay. So I added some lines just to illustrate that fact. This individual is deceased, and then these two individuals are deceased. Okay. Now, we looked at that one last time. Okay, how about this one? Dominant or recessive? Wrong. No, it's recessive. Yeah, it is. You know what? Here's what's confused you. This individual is not a member of the family. Whoa, what? I didn't write it. This brings up an important point. When you're looking at these, be very careful. This individual married in from outside. He's not one of the individuals that was produced from these two individuals, okay? So this individual is affected but produces no affected offspring. Out of 11, the probability of that is very, very small. All right? So that tells you that this is not a dominant trait. It is recessive. Okay? Do you ignore this? Well, yeah, if you're asking yourself the question, you know, does this trait appear every generation, you've got to ask every generation genetically, right? You've got to say this individual is not genetically related to any of these, okay? So that one came in from outside. So in a sense, you can ignore that individual for that purpose, okay? All right, is it X-linked or is it autosomal? See, I've confused you now. It's actually X-linked. Here's why. Here's why. This individual is producing no affected offspring. However, when you have this affected individual reproducing with this female who was derived from an affected individual, they're producing affected female offspring. She's a heterozygous carrier. He's affected. And having these two female offspring being affected is a pretty clear indicator that this is an X-linked trait. But if you're not sure of that, take a look at this. Here's two females that are unaffected but are producing unaffected or affected male offspring. All right? That's a clear indicator typically of an X-linked trait. So this is an X-linked recessive trait. Okay. Here's another one. Oh, yeah, good question. The question was, what's this triangle with a 5 in it? This indicates that there are 5 offspring from this mating, and the sex of those is irrelevant to answering the question. A lot of times you'll see this in pedigrees with large families, so they can just compress everything and say, well, these individuals were all unaffected. We don't really care what their sex was, so we just indicate that there were 5 offspring and just use a diamond. Okay. So if you're looking at this, it's present every single generation. It's a dominant, but it's a weird sort of dominant, isn't it? Okay, because it's not X-linked, because this male produces affected male offspring. So does this one. So does this one. All right, now your brain should be working at this point and saying, well, wait a minute. Males have that funny Y chromosome. And there are some genes that are linked on the Y chromosome. And what you see in those cases is transmitting from a father to every single son. And that's exactly what you see here. There is no son in this family that is unaffected. All right, so this is a Y-linked trait. And in those cases, we often don't even refer to dominant or recessive. We just say it's Y-linked. The reason we don't say dominant or recessive is because it doesn't make any sense. There's only one Y chromosome, so it's not competing against anything. All right, so this is a Y-linked trait. On that handout I gave you, you might want to add the Y-linked, and just indicate that if every male is affected, and you have transmitting from a male to every male offspring, it's Y-linked. Right? Very, very clear indicator. Right? Every male in the pedigree is affected, except the individuals that married in from outside. Okay, here's the last one we'll do. Excuse me. Dominant or recessive? Right. X-linked or autosomal? X-linked. X-linked. Here's why. You've answered the question dominant versus recessive. The next thing you do is you look at the offspring of male individuals. Here's a male who transmits the trait to every daughter and no son. Here's another male that transmits it to every daughter and no son. Here's a female who is affected who transmitted it to all of her offspring, but that's consistent with X-linked dominant traits. Okay? These males inherited her affected X, and so did this daughter. All right? But the real telling things are the offspring of these 2 affected males. That's what will clinch it as X-linked versus autosomal. Okay, so there's some examples of how you look at pedigrees. You sort of go through this mental analysis to try and figure out what's going on in those pedigrees. Any questions about those? Okay, good. I want to talk a little bit more about sex determination and differentiation and how the process actually occurs. In order to do that, though, I want to make sure that we're all on the same page, so to speak. So, is that individual male or female? Be careful. That individual is male. She's a he. Genetically, she's a he. Right? She has an XY set of sex chromosomes. If you were to do an MRI of her lower abdomen, you would find that there is a blind-end vagina. There is no uterus. The gonads internally are actually testes, not ovaries. Okay? But all the secondary sexual characteristics are clearly female. It's a defect called the TFM receptor defect, and as I go through this, you'll understand what that means and how it results in this kind of a phenotype. It's interesting. There have been a number of supermodels who suddenly found out when they failed to menstruate that they were, in fact, genetically male. Okay? They were XYs. XY TFM receptor defect individuals tend to have a more sort of voluptuous body. They tend to be taller than normal, that sort of thing. They tend to have a little bit accentuated secondary characteristics. Right? So it's not surprising. I'm going to dispel some rumors and some misconceptions, I think. It's long been said, and you hear this all the time, that human embryos start out female. They do not. They start out its. Okay? Or you might say they start out both. It depends on what part of the anatomy you're talking about, but they definitely do not start out female, although the female is the default state for the human embryo. The development of the gonads depends, the testes or ovaries depend on the presence of the X or the combination of X's and Y chromosomes. If there's a Y chromosome, then what ends up happening is this gonad develops as a testis. Okay? And what it does then is it does two things. It produces testosterone, which signals all of those primary sexual differentiation effects, the sex-specific plumbing, mostly, the development of the external genitals. If, however, the Y is absent, as in this upper part of this figure, what ends up happening is that that gonad develops as an ovary. Okay, so it starts to mature eggs. In a female fetus, what ends up happening is the ovaries are starting to produce all of those potential eggs. All of the cells actually start meiosis. Right? They go into prophase, and then they arrest in metaphase of meiosis 1. And they stop and they sit there for years, arrested in meiosis 1. It's one reason why older females tend to have a higher frequency of chromosomal abnormalities in their children. Things like Down syndrome. The frequency of Down syndrome goes way up as a woman gets older. By the time she's 40, she has as high as a 1 in 100 chance of producing a Down syndrome child. By the time she's 45, it's a 5% chance. Yes?
Is it also true that as the woman gets older, more eggs are dropped during menstruation, so their likelihood of twins is more?
I have no idea. The question was, is it true that as women get older, they mature more eggs so that they end up having a higher frequency of twins? I have no idea. Sorry. Okay. All right, so these eggs are arrested in meiosis 1, and they sit there for a long time. As far as the gonads are concerned, the other thing that happens is they produce estradiol, which is a female-specific sex hormone, which stimulates the production of the female-specific plumbing, the Mullerian ducts. Okay. The male-specific plumbing, the Wolffian ducts, will proliferate only if you have a testis. The testis also produces this stuff called MIF, Mullerian inhibiting factor, it's called. And that inhibits the Mullerian ducts. Okay? So the result of having a testis, because you're XY, you break down the Mullerian ducts. They don't grow. The Wolffian ducts do grow. In the female, the Mullerian ducts grow because estradiol stimulates them. And the Wolffian ducts that would give rise to the male-specific plumbing degenerate because there's nothing to keep them going. Okay, so here's what happens in terms of the excretory ducts. All right, so here's what I'm talking about. The embryo is indifferent. It is, in a sense, both male and female because it's got both sets of plumbing. However, one or the other of those is going to proliferate depending on the genotype. Okay, so we've got the Wolffian ducts, which are the male-specific plumbing, and the Mullerian ducts, which are the female-specific ductwork. If estradiol is produced from the ovary, that's in the XX situation, then the Mullerian ducts are retained and they develop, so you get the female-specific structures, anatomy. However, if the Y is present, then you get this Mullerian inhibiting factor that shuts down the Mullerian ducts and they degenerate. In the meantime, the Wolffian ducts grow because you've got testosterone available. So you get testes, the seminal vesicle, prostate, and all the other plumbing, anatomical plumbing that's necessary for the male. Okay? So it is not true that an embryo or a human embryo starts out female. It starts out as both or neither, depending on whether you look at gonads or excretory ducts. Okay? External genitalia develop similarly based on the presence of the Y chromosome. And it's all due to a response to the presence of testosterone. Right, so the testosterone is the critical factor there to get all of the male-specific genitalia. Okay? Again, the fetus starts out pretty much indifferent. It's got what's called a urogenital sinus, which will eventually form the excretory opening. Okay? But you get two different things that are happening based on whether you have the presence of an ovary or testes. Okay? The testosterone, or its derivative dihydrotestosterone, causes the production of the scrotal sac that's going to hold the testes. Okay? And then the penis grows as well. Notice the urogenital sinus in the male ends up sort of migrating up and closing off. There are individuals that are born where the urogenital sinus hasn't closed off all the way. It's a fairly minor problem. Okay, so lots of different things are happening based on whether you have the Y chromosome or not. Okay, so what we can do then is we can look at a time course and ask what periods in human development do these events occur, and what kinds of things are happening. All right, so from 0 to 6 weeks, we don't have a whole lot of stuff that's going on. The indifferent gonad develops, the gonad that will become either a testis or an ovary. Okay. The real critical decision here is made about week 7, which is pretty early. All right. The embryo determines whether there's a Y chromosome present in its cells or whether there's a Y absent. Okay. The presence of the Y is critical because of one gene in particular on the Y chromosome, which is called TDF, the testis determining factor. Okay? Testis determining factor. If that gene is present, then the individual develops testes. And now here's the really funny thing about this. You can have situations where that gene has gotten moved over to the X chromosome. So you can actually have XX individuals that develop as males because they have the testes determining factor. It just got moved, translocated onto the X chromosome. All right, so really we're not even saying that you have to have a Y present. You just have to have the TDF gene present. If you've got that, you're going to develop as a male. That's what determines maleness. So if you have that gene, then this gonad develops as a testes. If it's absent, it develops as an ovary. Right? What is TDF? Well, it's a protein that turns on or shuts down other genes. Okay, so it controls the activity of genes that determine male characteristics. In this case, the development of the testis. And all that's happening at about 7 weeks of development. All right, if you have a testis versus an ovary, then different things are going to be happening here. All right, if you have a testes rather than ovaries, you produce testosterone. That causes the Wolffian ducts to develop. You also produce MIF, Mullerian inhibiting factor, and that causes the Mullerian ducts to degenerate. Remember, the Wolffians are the male ducts, the Mullerians are the female ducts. So what's being achieved here is that since that individual is developing as a male, it needs to get rid of the Mullerian ducts, and it needs to keep the Wolffian ducts. So the testosterone stimulates the Wolffian ducts to develop. The Mullerian inhibiting factor causes the Mullerian ducts to degenerate. If the Y is absent, then you have an ovary, and there's two consequences of that. The ovaries produce estradiol, which causes the Mullerian ducts to develop. And since there is no testosterone, the Wolffian ducts degenerate. There's nothing to stimulate them to continue developing, so they break down. And all those cells die, and that material gets reabsorbed. This is happening between 7 and 16 weeks of development. Okay, so the testis versus ovary decision is an important one. Another thing is important in this process. Okay, testosterone is acting through a receptor protein, a protein that binds to testosterone and acknowledges, in a sense, chemically, that testosterone is present. So you must have testosterone receptors. Functional testosterone receptors to be able to maintain those Wolffian ducts so that they don't degenerate. Those testosterone receptors are very important for all those secondary sex characteristics that happen as well. Right? When males hit puberty, testosterone is flooding their system, but that testosterone is binding to receptors that cause cells to adopt those secondary sexual characteristics if it's necessary, those secondary fates, in a sense. Okay, so if you don't have functional testosterone receptors, you're not going to develop as a male. You're not going to start developing facial hair and thicker body hair and all that kind of stuff. All right, so the result of this is that we end up, if you have testes, because of the testes determining factor, because of a single gene, you're male. And if you lack that single gene, you're a female. There is a significant amount of brain imprinting that occurs after all of this has happened. Once testosterone is present in the system, or estradiol is present in the system, and the developmental decisions for the plumbing have been achieved, the brain starts to develop. Okay? And the brain develops along a male or female pathway. There really are anatomical differences between male brains and female brains. There are some things that females do much better than males, things like spatial perception, perceiving 3 dimensions. It's why females actually don't get lost as often as males. Because they can perceive 3 dimensions better. They use landmarks better than males do. Okay? Males aren't as good as that at that. So there really are some anatomical differences that control that. There are also portions of the brain, it's been found, that control sexual preference. There's a very specific portion of the brain that controls how an individual is attracted to other individuals. that region is smaller in females than it is in males. Okay? It's also, in homosexual males, it's the same size as in females, which is really interesting. A lot of brain imprinting that's occurring here. The basic innate behaviors are being set up here. Okay? It's why an individual who is genetically XY and has testes, but develops as a female, adopts a female behavioral pattern, because that's how the brain was imprinted. Okay, now we can go back and ask the question about this individual with the receptor defect, the TFM receptor defect, testosterone receptor defect. That's what this is. This individual lacks testosterone receptors. All right, so what happens if that's the case? The individual has a Y. They have the testes determining factor, so they get testes instead of ovaries. Right? So that part works. They produce Müllerian inhibiting factor from the testes. So the Müllerian ducts degenerate. That's why they have a blind-in vagina. They didn't develop any of the female-specific plumbing. There's no uterus, there's no fallopian tubes. Okay? The testes are in the same place that ovaries would be, except they're testes. But you have to have a testosterone receptor, have to have functional testosterone receptors to be able to respond to produce and maintain the Wolffian ducts. So since their cells couldn't respond to testosterone, blammo, there go the Wolffian ducts. Right? They don't have any of the internal plumbing, sexual plumbing that's necessary for either the male or the female state. Because their cells fail to respond to testosterone, they don't develop external male genitalia. Therefore, they go through the default state, which is female. That's why they have female genitalia. And their brains get imprinted in a female-specific behavioral pattern. Therefore, they behave as females rather than males, and they are attracted to males rather than females, typically. Okay? I guess the reason that I talk about this is that it's interesting that there are a lot of misconceptions about this process that can be cleared up. It relates back to what we've been talking about with sex determination and sex differentiation the last few or several times. Okay, but also it's important to realize that and I'm probably going out on a limb, and I'm probably going to piss some people off, but I don't care. I think it's very, very premature to label some behaviors as a choice when, in fact, they may be genetically controlled to a great extent. It's this part right here that we don't understand well enough to make any conclusions about yet. Right? But there's some tantalizing evidence that suggests that the way that we perceive other people starts out right here, and to some extent it may be unchangeable. Okay? Interesting things to think about. All right, well, we'll stop there, and Friday we'll go through some more problems just so you have a good handle for the exam. And I'm also wanting to do evaluations Friday, so make sure you bring a number 2 pencil to class so you can fill those out.
When we first started doing those, we had circles for the carrier. We had circles with color just inside.
Yes.
Was that